Welcome to the next lecture of our course on environmental biotechnology, and in this particular lecture we are going to discuss about some of the important case studies on bioremediation. So, in particular these two aspects of bioremediation, that is the bioremediation of chromium and bioremediation of uranium will be discussed, particularly with reference to the case studies where the bioremediation processes are being implemented in today's lecture. Now, before we go into the details of these two case studies with respect to bioremediation of uranium and chromium, let us quickly have a kind of a recap of the bioremediation process. And as you might recall that we already discussed in detail about these aspects, that in most of these all cases the bioremediation processes are actually catalyzed by the microorganisms, and these microorganisms are capable of transforming different contaminants. So, we have these contaminant, in this case let us say the toxic chromium 6 plus or uranium also 6. So, whether it is the contaminant, toxic contaminant like chromium or uranium or maybe the petroleum. So, whatever may be the contaminant, or maybe even the plastics also or other hydrocarbon HCs. So, the microorganisms which live in the natural environment, they are capable of transforming and interacting these with these toxic contaminants. So, the basic concept again is quite simple, that the microorganisms they interact with these toxic contaminants. So, if we try to keep our focus on the toxic metals and radionuclides like uranium, then we might be able to identify that these toxic metals like these metals, they are getting interacted by these microbes, because the microbes they are having a negatively charged surface because of these cell wall and cell membrane components, and some of these divalent cations etcetera, where they are being sorbed or accumulated inside the cells. A major process is the redox mediated transformations. So, these transformation reactions could be oxidation reactions or could be the reduction reactions. So, basically the metals are oxidized or reduced. So, during this case studies scenario, we will see that how these processes are actually being implemented. So, we just have a quick recap again of these microbial processes, where we see that the microbes are able to either accumulate or sorb the different divalent cations, and including some of the anionic complexes like the chromate or the arsenate etcetera. And the another very important aspect of the metal microbe interaction that I was referring to earlier, that is the biotransformation or the reduction based processes, and these processes could be reduction or could be oxidation as well. So, there are processes which are actually bio-oxidation or bio-reduction. The similarity of both these processes, that is the bio-reduction and bio-oxidation, the similarity is that in both the cases the oxidation state of the metal like chromium, arsenic, uranium etcetera, the redox active metals, their oxidation states are altered. This alteration is basically done because either these metals or metalloids are used as source of electrons. So, they are oxidized, or they are used as an electron acceptor. That means, during this process of electron accepting, as you know that the microbes are capable of using different metal ions as inorganic electron acceptors. So, they are able to perform their anaerobic respiration using these metal ions. So, these reductive processes or these oxidative processes are both found to be very important for the bioremediation purposes. There is another process which is also found to be very interesting and gaining importance is the biomineralization, where the microorganisms they release a number of ligands like the phosphate or H2S, as you can see the specific group of microorganism. For example, the sulfate reducing bacteria, for example, they produce the sulfides, and these sulfides could react with the metal ions and the metal sulfides are precipitate. Alternatively, the phosphates ion which are produced by the many bacterial strains using external phosphate organic phosphate donor molecules. These phosphate groups are also able to interact with the positively charged metal ions, and then they produce the metal phosphate complexes. So, it is basically the these two types, that is the redox transformation based processes and the biomineralization based processes. So, before I go into the details of this process, I would like to quickly have a recap further detailing of these processes. For example, if we want to just see how these redox transformations are actually catalyzed. So, we can understand that there are bacterial strains naturally present. So, under any aquifer or contaminated environment, so microorganisms are naturally present. So, there are two important requirements from the microbial point of view. One is the electron source. So, you need an electron donor, and on the other hand you need an electron acceptor, and this is basically we called as terminal electron acceptor. So, one way you have to have some compounds or some inorganic material, in this case we are talking about metals and radionuclides. So, you have to have something which will be donating the electrons, and these electrons will be processed through the diverse metabolic pathways and eventually will help the cell to produce the necessary ATPs and reducing power, and ultimately these reducing powers will take the electrons to the electron transport chain, which is located in the membrane. Now, this is actually the electron transport chain. If you remember, this is composed of a number of electron carrier complexes, and these electrons which are coming from this reducing power, which is like NADH H plus or FADH2 donating the electrons to this electron transport chain complexes, eventually capable of transferring this electron to the terminal electron acceptor. So, we call them TEAs, terminal electron acceptors. So, under the aerobic condition, for example, it is the oxygen who acts as the terminal electron acceptor, and oxygen is reduced to water. However, under anaerobic condition, compounds like nitrate, Mn4 plus, iron, sulfate and even carbon dioxide, they can also accept the electrons, and then they produce the reduced product, like nitrate can be converted to nitrogen, Mn can be converted to Mn2 plus, Fe3 can be converted to Fe2 plus, sulfate can be converted to sulfide, and carbon dioxide can be converted to methane. Now, these are the situations where we have the anaerobic condition. Now, depending upon the environmental situation, like in an aquifer environment, now we know that the aquifer may be aerobic, may be partially aerobic, it may be having a fluctuating oxygen level, or it may be anaerobic. Similarly, a contaminated soil or contaminated landfill or any other contaminated sediments where lots of metals and other pollutants are there, they might be having the aerobic or anaerobic conditions prevailing in them, and in between you have always a kind of a transition zones where the concentration of oxygen is gradually declining and the microbes are able to metabolize the alternate electron acceptor. This is number one, point number one. Now, when we have the pollutants, so we are talking about the pollutants, in this case we have the metals. So, some of the metals can act as E donor, while some of the other metals can be act as terminal electron acceptors. Obviously, under not all redox conditions both these processes will be applicable. So, some of the metals will be able to donate electrons under certain redox condition, whereas some of the other metals will be able to accept the electrons under the same redox potential condition. So, if we look at these two toxic metals, that is the point of interest in today's class, that is the chromium for example. So, we are going to talk about hexavalent chromium for example, and the uranium. So, uranium 6 plus, we are talking about uranium 6. So, both these are able to accept electrons. So, under the prevailing conditions, so it has been found that many bacteria, it is not the property of one particular group of bacteria, but many groups of bacteria we see that they are capable of utilizing this chromium or chromate or uranium, which is basically uranium 6 plus, as uranium 6 as electron acceptor, and they use diverse mechanisms to reduce this chromium 6 to chromium 3. So, it will be reduced to chromium 3 and uranium 6 will be reduced to uranium 4. So, that is the point number 2. So, under anaerobic condition especially, although some bacteria can do these reduction processes under aerobic condition also, because they have the enzymes which are capable of doing such functions. So, these processes are actually implemented during the bioremediation processes, which could be in situ or ex situ. So, most of the time these in situ processes are preferred, because in the in situ process the waste material like the aquifer, the groundwater, the sediment, the soil is there in place. So, the remediation process will be adopted while the toxic water or the toxic waste material is in place. It is not going to be removed from its original location. Now, let us first look at this case study. We call it field study 1 for aquifer chromium bioreduction at Hanford site using a polylactate biostimulation approach. So, this biostimulation approach is a very interesting approach, where we see that if you try to recollect some of these bioremediation strategies, that indigenous organisms, that is the native microbial community often possess the relevant organisms. These organisms mean multiple species or multiple populations. So, when you may expect that the community itself is having relevant organisms. These are different members of different genera, members of different populations, etc. Now, in a contaminated aquifer environment, you might have these relevant organisms. If you have these relevant organisms, then we can take up one strategy, that is called the biostimulation strategy. So, what is biostimulation? So, we add external agents which actually go diffuse and act on these relevant organisms and encourage them. They allow them to proliferate more, they allow them to function more aggressively, and in a simple sense, a simple sentence, that the relevant organisms are enhanced. So, this biostimulation can be done by adding sometimes electron acceptor, sometimes by adding electron donor, sometimes by carbon sources, or even can be by adjusting the pH or the redox conditions also. So, this is found to be one of the best strategy in case of a engineered remediation. So, when we say engineered remediation, you must be able to remember that there is another process which is called natural attenuation. In case of natural attenuation, we completely rely on the catabolic abilities of the native bacterial populations, and we do not interfere with respect to adding any stimulants like the biostimulation or something like that. Of course, there is another strategy in terms of the engineered approach like biostimulation we discussed, that is the bioaugmentation. So, in case of bioaugmentation, what we do? We add catalytically more active organism. So, often it has been found that the natural community is not having sufficient number of the relevant catabolically relevant organisms in this particular contaminated environment. So, we need to recruit organisms from external resources. So, we might have some cultures which may be mixed culture, might have some pure culture bacteria which are not ordinary bacteria, they are catalytically found to be more superior or they are more effective, efficacious in terms of the particular reaction that is being targeted. For example, if you want to have a chromium bioremediation, for example, we should have in mind that the chromium should be or must be remediated through reduction, that is the hexavalent chromium needs to be reduced to trivalent chromium. So, we should catalytically active organism means the chromate reducing bacteria simply. So, we should have some bacterial culture, some bacterial strains, and we can adopt that strategy as well, that is called the bioaugmentation. In case of biostimulation, we do not add any bacteria directly, rather we add some stimulating agent as I mentioned earlier. So, during this field experiments which was done under the United States Department of Energy bioremediation program. So, a number of radionuclide and metals and metalloid contaminated environments were located, sites were located in the US DOE program, and basically these were all targeted at the different facilities which are called US DOE nuclear production facility, using a combination of hydrogeological, geophysical, geochemical and microbiological methods. So, a large group of scientists were engaged to perform the survey of a large number of sites, although we are going to talk about only one particular site in this case study, that is the Hanford site. And let us go to the site. So, this is the picture of the Hanford site. As you can see, this is perhaps the one of the most beautiful picture of this Hanford site nuclear reactor facility, which is built in order to produce the weapon grade nuclear material. And I have learned that actually the atom bombs were actually having the plutonium which was enriched produced in these reactors, this Hanford site reactor. So, historically it has a great importance. And this particular picture seems to be taken in 1960, and the fact that is relevant for environmental aspect is it is just in the bank of the Columbia River, and we can see number of nuclear reactors. So, these are not operating anymore, and what is important is that these reactors and the associated facilities, including the storage of the spent fuel and all those things, they produced huge amount of waste. So, if you look at this Department of Energy facility produced nuclear material at the site for more than 40 years, and chromium was found to be one of the most important toxic material which is later discharged into the environment. And this chromium, though it is not radioactive and as such the nuclear program might not have a direct connection, but it has certain obvious reasons to be there, like is to prevent the corrosion in the cooling towers at the site and as an oxidizer in the nuclear fuel production process. And consequently, the site has a large plume of concentration of chromium 6, which is highly toxic, more than 2000 ppb, and in some studies it is referring to up to maybe 4000 ppb, and that is impacting the Columbia River. That means, they have some underground plume that we can see over here. So, this is a cartoon published in one of the environmental science and technology papers by the group of scientists who was working on in situ redox manipulation process. That was another process developed through permeable reactive barrier in order to combat the flow of toxic chromium. So, as you can see that underground, So, at the underground of this there is a plume of chromium contaminated water, and this chromium contaminated water is flowing towards the river. So, gradually this chromium contaminated water is coming into this river and contaminating the river and putting the entire river ecosystem under severe threat and affecting the public health, drinking water system, the overall ecosystem balance. So, USDOE has taken severe measure to combat this situation, and along with the chemical and physical and other processes, the bioremediation events were also taken up. Now little bit more background information about the chromium contamination. The chromium in its hexavalent form is a potent toxin, mutagen and carcinogen, that is a common groundwater contaminant at industrial site worldwide. So, no matter whether it is a nuclear facility or a non-nuclear facility. So, chromium, hexavalent chromium is a very commonly used industrial chemical, and it is often found in many of the industrial waste contaminated sites, including India. Now, it has two commonly observed oxidation states, that is the point of interest under relevant natural conditions. So, it has two oxidation states which can be observed under natural conditions. Now, the most common chromium compounds in groundwater contain the hexavalent chromium or chromium 6 and the trivalent chromium, that is the chromium 3, which have quite different characteristics and properties. So, this chromium 6 and the chromium 3, they are quite unique. Unique in the sense they are both chromium and they are interconvertible, because chromium 6 can be reduced to chromium 3 and chromium 3 can be oxidized to chromium 6. Now, chromium 6 is a mobile element. So, it can, or compound, it can chromate ion can move very quickly, and it is very toxic in when it is in the groundwater or in the soil system. So, it is more soluble rather in aqueous phase, present in alkaline and strongly oxidizing aqueous environment as chromate ion mainly, and also as bichromate complexes. So, overall the chromate and bichromate, all these are chromium 6 complex, and it is very mobile, present significantly in the aqueous medium, and also it is bioavailable and it is very toxic. In contrast to that, chromium 3 complexes are less toxic, and it forms insoluble and stable precipitates under slightly acidic to neutral conditions in aquifers. So, what does that signify? It signifies that chromium 6 is very much soluble. So, let us take for example, if we try to just quickly look at the scenario, that just let us assume that this is underground situation and you have, this is the depth. So, you have the water inside and this is the sediment part, this is all the sediment, and in between you have this is the aquifer for example, and you have otherwise all these are different layers of sediments and rocks. Now, within this you have the chromium 6 plus. Chromium 6 plus therefore, will be in the aqueous phase. So, this is the water phase and this is the solid phase, or the solid or the rock or salt, sand, sediment etc., under the aquifer environment. Now, the moment you convert this chromium 6 to chromium 3, now chromium 3 cannot stay in the aqueous phase. So, basically it forms insoluble complex with different complex with the present with the available ligands, and it precipitates, and the precipitates are quite stable precipitate. That means, the precipitates that those are formed will not be or will not readily oxidize back to the chromium 6, not readily. So, particularly under slightly acidic to neutral conditions, and if you look at this, the most of these aquifers that we are dealing with are mostly circumneutral conditions. Now, owing to the low solubility, and mobility of this reduced chromium in groundwater, because as I explained that once the hexavalent chromium is reduced to chromium 3, the chromium 3 is having a low solubility and mobility, and it tends to always precipitate and form stable complexes with the this sediment and other solid phases of the environment. Now, in situ reductive immobilization of chromate plumes, that chromium containing the groundwater system, is considered an attractive remediation strategy. So, in underground system, if we can reduce chromium 6 to chromium 3, the chromium 3 will precipitate, and that precipitated chromium 3 will help us to get the water free from the soluble chromium. Now, this chromium 6 can be reduced microbiologically, of course, can be reduced chemically also. So, we can think of chemical reduction as well as biological reduction. Now, in environmental biotechnology, this reduction of the chromium through the bacteria in particular is highly important. So, microbially mediated chromate reduction has been reported under a range of conditions, under a range of electron accepting conditions, because in underground aquifer we often see a mixture of electron acceptors are there, and chromium might need to face a competition with respect to accepting the electrons, because many other electron acceptors might be there. And since other electron acceptors are also there, like iron, sulphate, nitrate, etc. are there, some of these which produce the respiration products such as the Fe2 or hydrogen sulphide, that increase the probability of secondary abiotic chromate reduction. So, what is that abiotic chromate reduction? So, we have already seen that there are microbes who are capable of reducing chromium. Now, under the aquifer environment, chromium is not alone. So, we have the chromium, and along with chromium, as I mentioned, that we have. So, chromium is there, but along with chromium there are nitrate, particularly iron and sulphate. So, when we have these microbes working on them, so they may reduce chromium, they may reduce nitrate, they may reduce iron, or they may reduce sulphate. So, if they reduce iron, for example, they produce Fe2 plus, or if they reduce sulphate, they produce H2S. Now, interestingly, this Fe2 plus can be reoxidized under the aquifer environment with reducing chromate 6 plus to 3 plus. So, instead of direct microbial or bacterial reduction of chromium 6, Fe2 can be aiding into the reduction of chromium 6 into chromium 3, and similarly H2S can also do that. So, H2S can aid into the reduction of chromium 6 to chromium 3, forming a sulphate. So, these are also possible. So, direct reduction of the chromium is possible, as well as the indirect reduction of the chromium is possible. Now, what are the direct and indirect? Among the common abiotic reductants of chromate, the hexavalent chromium, the iron is found to be most efficient, Fe2 plus. And it is also very interesting that under the aquifer environment, Fe3 is abundant as a part of the geological properties. Now, this Fe3, if it is reduced to Fe2 by microbial action, this Fe2 can act towards reducing chromium 6 further. And in that regard, scientists have done a number of studies, and what they have observed, that not only Fe2, there could be other chemical compounds are produced. For example, under the range of conditions, that is a pH 2 to 9, with environmentally relevant concentration of the potentially reductants, including the Fe2, hydrogen sulphide, which can be produced very well by reduction of iron 3, Fe3 plus, or the sulphate, fulvic acid, phenol, magnetite, etc. In most of these cases, scientists have found that Fe2, which could be easily produced by reducing Fe3 plus, is kinetically more efficient, and it kinetically outcompeted the other potential abiotic reductant. That means, even if you have this fulvic acid, phenol, magnetite or sulphides, the Fe2 will be a potent reductant for reducing the chromate. Now, during this in situ approach for this Hanford site, initially the in situ remediation of chromium contaminated groundwater was done involving both chemical as well as biological agents to reduce the chromium 6 to chromium 3, which is actually the chromium 3 is obviously, as I mentioned, is less toxic, less soluble and less mobile than chromate. So, the target was to produce or the convert chromium 6, which is very hazardous, to less toxic and stable precipitate forming chromium 3. Now, chromium 3 can precipitate as hydroxide, usually as a solid solution with ferric iron oxides, and can be effectively immobilized. So, that is a part of immobilization that can be achieved, particularly when we have iron in that. Now, this reduction is usually a permanent solution, because chromium 3 is not easily reoxidized to chromium 6 under conditions that occur in the most natural groundwater environment. So, the reverse reaction is possible, but not feasible under the conditions which are prevailing in the groundwater conditions. In most of the cases, scientists have studied this thoroughly and come to the conclusion that under the natural groundwater condition, the reverse reaction, that is the oxidation of chromium 3 to chromium 6 is not preferred. Now, the chromium reduction is commonly used chemical reductant for chromium 6 to chromium 3 is chemical reductant with subsequent immobilization to of the chromium 3 is the Fe2 plus, as I mentioned, because it is found to be kinetically is much superior than other. Now, this particular approach was taken in the Hanford site as well. So, the Pacific Northwestern National Laboratory PNNL USA has developed and implemented an in situ redox manipulation technology, that is called ISRM technology, using the injection of sodium dithionate solution into the groundwater. Now, what is the job of the sodium dithionate? Sodium dithionate injection into the aquifer leads to the reduction of Fe3 releasing Fe2, which is needed for the chromate reduction. So, dithionate is producing the electrons, and these electrons are capable of reducing the Fe3, which is present as ferric oxide or hydroxide in the aquifer solid phase, and this resulting Fe2 is capable of reducing chromium 6 to chromium 3, and Fe3 is getting produced. And this sodium dithionate is not a very stable compound. So, after some time this action of the dithionate will be diminished into the aquifer system. So, this was the report published in the journal Environmental Science and Technology, where in situ treatment of chromium contaminated groundwater was achieved successfully. Now, along with this and subsequent to this, the scientists found that why to inject this dithionate, rather in case of stimulating this bacteria, if that is the point that you want to actually have Fe2 plus, you are otherwise using sodium dithionite, but what about the fact that if we can produce this Fe2 plus in situ. So, the in situ bioremediation of chromium 6 was achieved, because previous studies demonstrated that simple organic carbon compounds like lactate could stimulate the iron reducing bacteria in the soil to reduce enough Fe3 to Fe2, that the Fe2 could reduce chromium 6 to chromium 3 and precipitate it in the soil. So, effectively the scientists they injected organic substances which are called compounds which are easily utilized by these iron reducing bacteria. So, through different injection wells they put these or injected these lactic acids or polylactate compounds, and these compounds are finally utilized by these iron reducing bacteria which are naturally present. So, these compounds might be available to all organisms. Of course, the iron reducing bacteria will be happy to utilize along with sulfate and nitrate reducing bacteria as well, but we are more focused on production of Fe2. So, Fe3 will be reduced to Fe2 by this iron reducing bacteria, and then the Fe2 will be able to reduce chromium 6 to chromium 3. Now, polylactate was finally chosen as a compound to be injected, and it is also called hydrogen released compound. The polylactate actually hydrolyzes to lactic acid in the aquifer, which is readily utilized by the indigenous bacteria, particularly the iron reducing bacteria. And basically this work was published in the journal Environmental Science and Technology with the title in situ long term reductive bioimmobilization of chromium in groundwater using hydrogen release compound. And this was the sampling or the injection well strategy, that you can see that you have the injection well, as well as the pumping and the monitoring was continuously being done in order to facilitate the process. So, what was observed that in 2 weeks time after the injection of this polylactate compound, the total bacterial diversity increased from less than 10 to the power 5 cells per ml to more than 10 to the power 7 cells per ml. So, enhanced bacterial activity. As expected, the oxygen was depleted fast, because in the presence of lactate, the aerobic organisms will try to consume the available lactate, and very soon the oxygen will be depleted, and then nitrate and iron will be reduced. Within 3 to 4 weeks time of injection, the chromium 6 came down in the monitoring wells and stayed well below the level of drinking water for several months, even after the nitrate and oxygen returned to the original concentration, because as I mentioned, the chromium 3 is a very stable form, and under the prevailing groundwater conditions, the chromium 3 is not readily oxidized back to chromium 6. Subsequent microbial community analysis by microarray data suggested that a large number of organisms, including the Pseudomonas or Geobacter, even the disulfovibrio, the sulfate reducers, and archaea were developed or improved. You can see with respect to time scale how these different organisms are at different depth. So, these are the different colors indicate different depth, and at the different time, like days post HRC injection is data is presented. And here is some of these screenshot of the good papers which might be of interest, and subsequently the long term effectiveness of this in situ immobilization as a remediation strategy was studied, because it was observed that it could be reversing back, like oxidizing back to the large extent the stability of the reduced chromium. So, stability of the reduced chromium was also studied, and it was found that the bulk redox status of the biogeochemical regime as categorized by the dominant electron accepting process do not necessarily control the final product of chromium 6 reduction. So, the overall study showed that there is a very high degree of implementability of this concept, and they successfully remediated this site. Now, next we will learn about the uranium bioremediation through the case study. Now, uranium contamination is again linked to these nuclear activities, which is considered to be a legacy of the environmental contamination. An elevated concentration of uranium and along with uranium different other radionuclides present in the mining and milling area at the sites where uranium ores are processed, and where uranium was enriched as a part of the nuclear processing, nuclear industries, including the nuclear warfare as well as the nuclear energy. And this contamination potentially represent an uncontrolled source of radiation also, and therefore, regulatory bodies may require it to remediate the acceptable level. So, that was the understanding from US DOE side. So, United States Department of Energy, they took adequate initiatives to actually control the problem with uranium and other toxic metal and radionuclide pollution. Now, with respect to uranium again, as we have seen with respect to chromium, the mobility of the uranium in the environment depends on the speciation and the oxidation state. Again, it has two very common oxidation states, one is uranium 6 and uranium 4, and in uranium 6 in oxidizing conditions predominantly is a form of urinyl ion UO2 plus or hydroxyl complexes, as you can see. So, with a pH regime close to 6 or below that, we can expect that it will be mostly that uranium 6 plus UO2 plus urinyl ion we call it, or it may form some urinyl carbonate complex at higher pH. So, as we move into the higher pH, it forms the urinyl carbonate complexes. So, essentially again the solubility and the toxicity of uranium is highly linked to this oxidation state. So, uranium 6 is highly soluble in aqueous medium, is toxic, whereas the uranium 4, which can be produced out of reducing the uranium 6, is very less soluble, and it forms complexes with the carbonates and other materials present in the aquifer solid phase. Now, the biogeochemical interactions have been found to be very important and playing a key role in controlling the speciation, that is the interconversion between the 6 and the 4 species oxidation state of uranium, and that is actually linked to the mobility of the uranium, and it is also true not for other toxic redox sensitive radionuclides like technetium, neptunium and plutonium also. So, direct, these are the two processes are found. One is the direct metabolic process, that is the microbial respiration. So, in case of uranium, there are increasing number of evidences that the microbes can actually reduce these uranium 6 directly, that is called direct respiration. There is another way of reducing it, that is called the indirect reduction, that is the changing the ambient redox pH conditions, producing ligands or new biominerals or altering the mineral surfaces. So, in geological context we can understand that when the microbes are able to produce these ligands or produce different biominerals or alter the mineral surfaces, the uranium 6 can precipitate heavily on those. So, for example, as I mentioned, the uranium 6 can be reduced directly, or microorganisms can produce certain ligands or certain groups, and these can actually bind to the uranium 6 and lead to the precipitation of the uranium in the solid phase. So, therefore, the in situ bioremediation of uranium within the contaminated groundwater sites can be achieved by two processes. One is the natural attenuation, as I mentioned earlier, via controlling the radionuclide mobility, and this natural attenuation is absolutely relying on the abilities of the natural microorganisms, microbial communities, or else biostimulation can also be done, like via activating the natural biogeochemical processes, the geochemical process which are going to actually transform the uranium 6 to uranium 4, or will produce the ligands, or will bring such changes in the solid phase or produce other molecules which might interfere with the solubility and will make the uranium precipitate into the environment. So, we have found that these are the 4 basic mechanisms with respect to microbial interactions with uranium. These are the uranium bioreduction is on the top of the list. It can be reduced by diverse mechanisms, and along with that we have the biosorption or bioaccumulation, but also interestingly this process which is biomineralization. We have already discussed these processes in detail in our earlier lecture on metal microbe interaction. With respect to uranium bioreduction, it has been found that this could be a potent bioremediation strategy. Now, what happens in the absence of oxygen under the aquifer condition? So, bacteria are able to respire with different other electron acceptors to gain energy for metabolism, because they need to utilize different electron acceptor, so that the energy is gained, because these electrons are eventually passed to the terminal electron acceptor, and the reducing power like NADH plus or FADH2 is reoxidized. So, electron transport chain facilitates this process. Now, as the anoxia progress, the most energetically favorable electron acceptor are used in a sequence, and that follows the redox chronology of redox reaction. So, if we consider that in aquifer environment we have some amount of oxygen, of course, some nitrate will be there, iron 3 and sulphate will be there. These are the common electron acceptors present in the aquifer environment. So, microorganisms who are living, so I am drawing one cell, but actually these are the job of multiple different cells, different species are active, because all organisms may not be active under the anaerobic condition. So, I am just drawing one cell here to show that these could be aerobic bacteria and utilizing oxygen under the conditions where oxygen is available. But as we go down and as we have some amount of organic materials, the oxygen is getting depleted. So, either you go down, you see there are diffusional barriers and other restrictions. So, the oxygen level also declines. So, as the oxygen level declines, the other electron acceptors like nitrate. So, nitrate will be acting as electron acceptor, producing back nitrogen or ammonia. So, there are different mechanisms. Similarly, Fe3 could be utilized an electron acceptor and Fe2 will be produced, and at strongly reducing condition or strongly negative redox potential condition, sulphate will be reduced to produce sulphide or H2S. And in further down, eventually we will see that carbon dioxide, which might be present due to the activities of these all microbes, because they are they are ultimately catabolizing different organic or oxidizing different carbon sources also, and the CO2 are produced. So, these CO2 molecules will be utilized as electron acceptor and eventually methane will be produced. So, this will be the chronology of electron acceptors which are to be produced. So, that will actually follow the diagram that we discussed earlier, and we can see that and depending upon these electron acceptors used, the energy gain will be different, like if they are using oxygen, the highest amount of energy will be gained compared to nitrate, and then the manganese or iron reducing bacteria, and then sulphate reducing bacteria, and finally, carbon dioxide reducing bacteria archaea rather. Now, this could be very easily observed in the aquifer environment. If we continuously monitor the dissolved oxygen level followed by the nitrate, Mn, iron, sulphate and carbon dioxide or methane, we will be able to see that they are following a chronology, like first the nitrate under anoxic condition, then the Mn, then the Fe, then the sulphate and then the CO2, because they are guided by the amount of energy which is produced out of this reducing process, because unnecessarily they are not going to reduce sulphate if nitrate is available, because reducing nitrate is going to give them more energy. Now, if there is abundant organic carbon in the aquifer environment, which are generally found to be oligotrophic, but if we have some arrangement that excess organic carbon is provided, then we may find out that nitrate and metal reduction or metal and sulphate reduction could potentially occur concurrently. So, the chronology may not be followed. So, the chronology that we have seen may not be followed. So, that means under these circumstances we can see that nitrate along with Mn are actually getting reduced simultaneously, whereas the iron and sulphate can also be reduced simultaneously. So, this is the beauty of the process. Now, particularly at pH close to neutrality, uranium 6 has a similar redox couple to Fe3, and Fe3 reducing bacteria are able to respire uranium 6 as an alternative electron acceptor, reducing it to insoluble uranium 4. So, if we go back here, we will be able to see that we have this redox potential data, and with the P values and also the redox couples are shown. You can clearly see that the uranium and iron, their redox couples are quite similar. So, the point is emerging over here that if you have sufficient organic carbon, for example, acetate or lactate, something like that. So, if you have sufficient organic carbon, so what is the job of organic carbon here, that we will see. So, the iron will be reduced. So, Fe3 will be reduced to Fe2, plus many of these bacteria which we called iron reducing bacteria. So, commonly they are called as IRB, iron reducing bacteria, they will also reduce uranium. So, uranium 6 will be converted to uranium 4. So, this reaction will happen, and this is true also for many of the sulphate reducing bacteria also. So, we will be able to see that it is not only that this IRB or iron reducing bacteria, they are capable of reducing uranium when particularly we have enough supply of organic carbon, but also the sulphate reducing bacteria, they are also, and some of the fermentative organism, they will be also able to join hands and will be reducing uranium to uranium 4, which is going to be readily precipitated. Now, uranium bioreduction has been proposed as a bioremediation technique stimulated by adding an electron donor to promote the enzymatic reduction of aqueous uranium 6 to uranium 4. Now, in the previous drawing, if we look carefully, what is the function of this organic carbon? So, basically the organic carbon is getting oxidized and it is providing the electrons, those electrons which are basically supplied to the electron transport chain via the electron carriers like NADHH plus or FADH2. So, the more amount of organic carbons which are readily metabolizable and metabolized by this iron reducing bacteria or even the SRBs and some of the fermentative bacteria, they are capable of reducing uranium no matter whether they are SRBs or they are IRBs. So, a number of strategies is actually established through different cytochromes, different nanowire, even extracellular released flavin compounds are found that they facilitate the transfer of electrons to uranium. Now, the case study that we are going to briefly see here is about the USDOE rifle site Colorado, and the groundwater as this former uranium ore processing facility. So, this is basically this was uranium ore processing facility, and in this facility we see that low level of uranium is there and which is leached from mill tailings into the unconfined aquifer. So, here you can see again. So, under this facility in the mill tailings where the mill tailings were dumped, the low concentration of uranium leached into the aquifer environment. Now, here also the similar type of strategy was adopted, that the organic substances like the acetate for example, along with acetate a large good range of compounds like methanol, ethanol, glucose, lactate etc., were tested in order to find their efficacy. And the basic logic was that to activate these iron and sulphate reducing bacteria who can actually reduce the uranium 6. Parallel studies microbial ecology and microbial diversity studies were conducted in order to identify these organisms, that what are these organisms present. So, it was found that subsequent to this process, so there could be actually events going on like this, that the once you actually inject this your acetate for example through the injection well, the indigenous microorganism of the contaminated sites are stimulated and using this organic carbon, and selected organism may couple the oxidation of organic carbon to reduction of uranium, because these could be the IRBs and SRBs which are then URB uranium reducing bacteria, and the reduced uranium which is uranium 4 may be reoxidized to uranium 6 following cessation of the organic carbon. So, there may be always a chance that uranium 4 can be oxidized back to uranium 6. So, in order to look into that aspect, the scientists have two options. One is the presence of diffusional barrier like a biomass or low permeability sediments, or a preferential reductant like iron sulphide complex. So, they have found that if we have this iron sulphide complex, so this iron sulphide complex actually allow the stability of this uranium 4 and maintain the immobilized uranium 4 within the aquifer solid phase. And the sequence of events were also identified, that in a stage A, stage B, stage C, stage D, how nitrate reducing, iron reducing, uranium reducing and sulphate reducing organisms, sulphate reducing bacteria are proliferating into the system. So, essentially this acetate utilization along with that the uranium reduction to uranium 4 was observed, and what was very prominent, that the growth of or the enrichment, natural enrichment of the Geobacteriaceae member, and in subsequent studies the microbial communities were monitored with all kind of tools including the culture independent transcriptomics, metatranscriptomics and genome sequencing. All these things were implemented. It was observed that in general Geobacter species have been found to dominate the microbial community during this iron reduction bioreduction, and many of these members they actually later identified as Geobacter urani reducens were found to have mechanisms, specific mechanisms through which they are able to reduce uranium. Now, whole genome microarray analysis found that the transcript abundance of RPSC, that is a particular ribosomal protein correlated based to the growth rate of Geobacter urani reducens. Therefore, monitoring expression of these could be a kind of a biomarker for monitoring the activities of these organisms. So, here are the screenshots of some of these papers which are related to the case studies of uranium bioremediation, and it is not only for the rifle site, multiple USDOE sites have been subjected to underground in situ bioremediation using similar concept. So, these are the references for general. In general, this first review paper seems to be very useful, along with other papers related to the chromium bioreduction and bioremediation case studies, and also the papers related to uranium bioremediation case studies. And in conclusion, the two case studies related to chromium and uranium remediation, bioremediation are discussed in detail. In particular, we see that how the injection biostimulation actually through injection of these carbon sources, the relevant microorganisms are enhanced or enriched naturally, so that they could perform the desirable function. So, thank you very much.